Donc, bienvenue à cette deuxième rencontre de la communauté d'échange entre scientifiques du domaine des sciences, des données et de la santé. Cette semaine, on a la chance d'avoir deux présentateurs. Donc, notre première présentation sera donnée par Julie Hussin, qui vient de l'Institut de cardiologie de Montréal. Et je laisse la parole à Julie.
Bonjour à tous, donc merci pour l'invitation. C'est un plaisir d'être ici et de vous parler de mon jeune groupe de recherche qui, en fait, je suis à l'Université de Montréal en tant que chercheur depuis la fin 2016, donc un peu plus de 2 ans maintenant. Mais j'ai fait mes études dans le programme de bioinformatique à l'Université de Montréal, donc c'est un retour aux sources pour moi. Et donc, aujourd'hui, j'aimerais vous présenter quelques projets, enfin 2 projets que mon groupe dans lesquels mon groupe est impliqué. I'm going to switch to English. It will be easier for me to talk about science, but feel free to ask questions in French, of course. So, before starting, I just want to present my research group. En fait, ça, je peux le faire en français. C'est mes étudiants. Donc, ça fait que 2 ans, mais on a grandi assez rapidement. Donc, ici, vous avez les noms de mes étudiants, maîtrise, doctorat, une postdoc, et aussi des étudiants du baccalauréat en bioinformatique qui sont stagiaires dans mon laboratoire. J'ai 2 bioinformaticiens dont le travail va être présenté aujourd'hui. En fait, c'est le travail que je fais avec eux, que je vais vous montrer. Et puis, on a aussi la chance d'avoir à côté de nous la plateforme du CDIC, qui est le Canadian Data Integration Center. Et la branche de Montréal est à l'Institut de cardiologie de Montréal, où nous sommes et nous faisons notre recherche. So I'm also an IVADO professor with the mandate of integrating and using data science in medical and health sciences. So I think it's a good place to present, given the title of this seminar. And so the idea, as you know, is that we are in an era of big data, and in all fields of science and of technology. And we want now to be able to integrate this in order to understand biological processes and health science. And so here what I'm presenting, many of you will know about this, but it's an extended version of the Dogma of Life, where you have DNA and DNA molecules and DNA variation, which is at the top of the dogma and leads to phenotypic variation in individuals, such as disease, for example, so healthy state, disease state of any disease you can think of, or any other phenotype that you want. And in between this DNA status and the phenotype status that we are observing in the population, we have all these other omics technologies that are now being interrogated by biotechnology. So epigenomic, which is modification of DNA that is not necessarily been inherited from generation to generation. Transcriptomic, which is the expression of the genes and of the genetic variation. Proteomic, which is the set of proteins and of peptides that are actually being activated in the cell, and metabolomic, which is the set of metabolites that are being used by proteins in the cells to then create these macrophenotypes. And all these phenotypes, we can call them endophenotypes, or molecular phenotypes, that are actually a link between the genetic variation and the phenotype traits that we are observing. And a big part of my research and my training has been focusing on looking at evolution and how we've got there in terms of why we have this genetic variation nowadays, and how that evolution has influenced the phenotypes. So this is a big part of my research, evolutionary medicine. It's a term that aims at explaining this field that attempts to explain human health through evolutionary thinking. And we're looking at, for example, the links between cardiovascular disease and fertility phenotypes, thinking about natural selection. We're also looking at natural selection in genes that are involved in pharmacogenomic interactions, so to understand how these different genes have been modulated throughout the history of humans. However, I'm not going to talk about this today, so this is just for your information. Today I'm going to talk about the cardiovascular omics project that we have in my lab and that are taking place in collaboration with clinicians and fundamental researchers at the Montreal Heart Institute. And I'm going to describe this project in this presentation. The third axis of my research is to try to develop new techniques, new bioinformatics techniques in machine learning for integrating this very wide array of different biological data we have, and I'll touch this at the end of my presentation. So these projects, the two projects I'm going to present are the transcriptomic of pulmonary hypertension, which is a phenotype that is related to heart attack, and the second one is metabolomics of myocardial infarction. And in each of these projects, we are also dealing with some drug interaction. So I'll explain the omic data that we have access to and the questions that we're trying to answer. But before that, I just want to do a quick cardiology one-on-one presentation, because I'm not a specialist in cardiovascular disease, and since the beginning of my group at the Montreal Heart Institute, I've been learning a lot from the clinicians over there and the fundamental researchers over there, and I came to the realization that before going there, I wasn't exactly sure what were the difference between these three conditions: heart attack, cardiac arrest, and heart failure. And it turns out that it's very important differences, and that in the general terms, heart attack and cardiac arrest are used interchangeably, but it's actually wrong. So you can say that the heart attack or myocardial infarction or acute coronary disease is more a problem related to plumbing issues, where you have a blockage of the artery, and the fact that no blood can go to the heart will damage the muscle, the heart muscle, locally, and this means that eventually this can lead to things like heart failure. Cardiac arrest is more of an electrical problem. It's the entire heart that stops. It can be a consequence of a heart attack, but it doesn't have to be. So this is a more serious kind of condition. And heart failure is actually the inefficiency of the heart to pump blood in the body. So it's linked to muscle damage, but it won't necessarily mean that the heart will stop. So it's just the heart not working properly in a way, and it will lead to different types of conditions. But so these three things are quite different. And there's also another component that we can add to this, is the fact that the pulmonary circulation is completely linked to the heart circulation as well. And so you can have, depending on the different conditions you can have also, when you have a heart attack or a cardiac arrest or heart failure, you can have damages to the to the lungs. And damage to the lung will then also lead to maybe right heart disease or heart failure. So all these things are linked together. And so this first project is a transcriptomic project of pulmonary hypertension of type 2. Type 2 pulmonary hypertension is the one that is followed, that occurs following myocardial infarction. So someone has a myocardial infarction, a heart attack and will develop potentially pulmonary hypertension, where the capacity of the lungs and the function of the lung will be reduced. And so one problem with this condition is that there's no treatment today to treat this type 2 version of pulmonary hypertension. But recently, Prometic, which is a Montreal-based company, had a new molecule tested for different conditions, including type 2 pulmonary hypertension, and it showed in different studies that it had benefits for treating this condition. And one study where Jocelyn Dupuis at the Montreal Heart Institute was involved with Prometic was to look in rats at what happens when you give the rats, so the rats have an MI, myocardial infarction induced, and then they gave the rats a drug 3 weeks after the myocardial infarction, and they saw great results in reducing ventricular hypertrophy and lung fibrosis when this treatment was given. And so then the next question was to see if you gave the drug earlier, so maybe 48 hours after myocardial infarction, would that give the same results? They were also comparing it with valsartan, which is a treatment that is not meant necessarily to treat pulmonary hypertension, but that is used in practice. And here you can see this new early study where they had 365 rats, giving them, inducing an MI. And you can see that 48 hours after MI, they are randomized into 4 different groups. The group without treatment, the group with the PBI 4050 molecule, the group with the valsartan, and the group with the combined treatment. And so at this stage, they wanted to do a lot of getting a lot of information on these rats phenotypically, so the cardio-phenotyping that they usually do, but they also had this idea of having a transcriptomics study. And so we designed, together with them, an RNA-seq study. So we sequenced the transcriptomic of 36 rats, split it in 6 groups. So you see them here. So the sham means that they had the operation that was done to induce the MI, but the MI wasn't induced. The MI groups mean that they got the MI induction. And then you have the different treatment that were given to them. And in each group, we have 6 individuals. So our questions here are, we want to know the effect of MI on gene expression, and we also want to compare the treatments. So here's the bioinformatics pipeline that we've used. It's actually the bioinformatic pipelines that we've used to produce the results I'm going to show you. But it's not necessarily the only one we tested, because there's lots of tools out there. But so here you see the RNA sequencing experiment that was done at the Genome Quebec Center, McGill and Genome Quebec Center for Genomics. You have, so then we use Callisto, which is a RNA-seq program that aligns all our sequencing data to the transcriptome, the reference transcriptome of the rat. So here it's the reference to the reference genome of the rat. Then we have a limit tool that corrects the data in some sense and gives us the total number of genes that are expressed in our data. Then we go to a lot of different steps of QC to make sure that the data is sound. And in the end, we do analysis, of course. So I'm going to go through these steps, except for the QC. If anybody has any questions, I'm just going to say that we do some dimensionality reduction analysis, and we use these kinds of technique to spot outliers. We looked at Excel program that helps quantify an enrichment for different cell types, because here you have to know that this is a bulk sample, so we're not selecting some specific cells, and then we are looking at the number of reads mapped and so on, and in our different groups. In all those slides that I'll present, you will notice that I excluded this sham plus PBI group, and the reason why is that it actually looks a lot like sham saline, and so I'm only considering one of these two. So the main picture you're going to be looking at is what is called a Volcano Plot, and so it comes from the fact that we're looking at the effect here of MI on gene expression. We're comparing the sham group to the MI group without treatment as first. And so the sham group is the reference, and we're trying to understand what are the changes in the MI group that are driven, that are seen in our sample. We're using a package that is called Lemma. It's a linear model framework that will evaluate the contrast between the two groups. And so the idea is, we here implement a design. So in this case, for example, the design will be the two conditions we're testing, so the MI versus the sham. And we obtain a bunch of different statistics, but the ones that I'll be talking about are the log2 fold change. So it's an estimate of the fold change, so the amplitude of the contrast of the effect in the group that is MI. So here, for example, if you have a large fold change in that direction, it means that it's overexpressed in that direction. So it's overexpressed in the MI group. And if you have dots here in that direction, it means that they're underexpressed in the MI group. So we transform it to have it in log2. And we also here have the minus log p-value, which is a representation of the significance of the effect we have. And this is called the Volcano Plot because here in the middle you don't expect to have any dots, because the more significant you are, the more likely you are to have large effects on either side. So here I'm showing the same plot. It's exactly the same, but I put colors on it just so that you see what are actually the genes that we consider to be significantly differentially expressed. There's two criteria. The first one is the significance. As you see, we get a threshold here for the significance. And the second one is the fold change. So this talks to the difference between statistical and biological significance. Although something can be highly significant in terms of the p-value if it's not a high fold change, and here by convention we use that it needs to kind of double the expression in one group or the other. We consider this as biological significance. This is something that is debatable, whether this value, this threshold value, is actually the right one to use. The second thing to consider here is the multiple testing issue. So the fact is that we are looking at 14,000 genes, and so we're doing a lot of these tests, and so we need a method to see what is the actual threshold that we trust to be significance in the view of all these tests. And so this is why we are using this Benjamin and Oshberg method. It's an FDR method. I can get into in more details if you want, but we're using this as false discovery rate will be 1% in our subset of genes that we are identifying. And so here we put a bit of the names of those genes. So maybe to you, except if you're in the field, you might not know what these mean. We didn't know what it meant when we looked at this either. But what we know is that we are finding interesting sets of genes that are at different FDR levels and at different amplitude effects, potentially interesting to look at to understand the signature of MI in the lung tissue. So here I forgot to mention that I'm looking at the lung tissue, and we have also looked at the right ventricular tissue. So we have for each individual 2 tissue. And so as you can see here, we had nothing really interesting in terms of the transcriptome. So you see a negative control in a way. It can give nothing. So up to now, I'm going to stick to the lung tissue. And in fact, going back to the clinicians, it was expected that we would see most of the things, most of the transcriptomic signal from the lung tissue, which was a good news for us. So then we can start looking at the different treatments. And again, here, so now we're comparing it to the reference that has changed compared to what I was showing you before. Here, the reference is MI without treatment, and then we're comparing it to the treatment every time. And so this is the result of PBI not doing anything significant on the transcriptome of these rats. If we look alvarzartan, it's the same result. Nothing significant comes out of these 2 treatments, which might be the fact that we have a small sample size, although if we look at the combined treatment, all of a sudden we've seen several genes being up and down regulated compared to the MI without treatment sample. That was interesting finding, and then we can compare these 2 volcano plots. So here, so before we were comparing different sample groups, and now we're comparing comparison. And so here we plotted the most significant, the 24 most significant genes in our 2 comparison. And as you can see, and here are the log to fold change of those 2 comparison. And as you can see, they're completely they correlated. They're in the opposite direction, so that it means that the genes that are upregulated here are downregulated here, meaning that probably what's going on here is, given the reference of the MI without treatment, we're first having those genes being up or down regulated, and then after treatment, those genes are coming back to their steady state status, which means that we're kind of identifying some pathways through which MI has been that modulate MI or are a consequence of MI, and then given the treatment, we're going back to the steady state. That was good news to our collaborators, because they already knew, they didn't tell us, but they already knew that the group that succeeded in terms of the phenotypes and the echocardiographic parameters were the combined treatment. So this is the one at the end. Here you have the MI without treatment. So those are the sham. Those are the MI without treatment. And as you can see, for example, for the right ventricle global activity performance, this category is in between those 2. So it kind of mean that it's not as bad as MI, but it's not yet as good as sham. And all these parameters, so here you have the left ventricle performance, you have a pulmonary artery measurement and the ejection fraction as well, so measurement of the pulmonary activity and the heart activity. So then, once we have this overview, once we have discovered that this treatment is probably the one that has the highest benefits, and that also the transcriptome is moving in that direction, we can start looking at the overall transcriptomic functional differences between those groups. Here are all the genes, so we consider all the genes that were significant in at least one of the comparison we did, and we can derive this heat map where you have a value that represents the enrichment or the depletion of the genes depending on the different samples. And you can see clearly that there's two groups of genes that are moving together in the same directions. And in terms of the clustering of the samples, we can see that some of the sham here are clustering, and we have an enrichment for also the combined treatment on the left of the graph. We can use another type of approach to look at this. It's a spanning tree approach, a minimum spanning tree approach, where in fact we are trying to create a graph that has as few as we can edges, no cycles, and so in fact it's a tree that links all the samples together with the minimum distance. And what we are seeing here is that the sham and the combined treatment are clustering together. We have this individual here that seems a bit like an outlier, but it happens. And then we have all the other MI that are clustering together on this tree. We can also do pathway analysis using, for example, a tool that is called Progeny. Progeny is a R package that infer pathway activity from gene expression data. It has been used mainly in cancer, but it's actually useful in many other application for sure. And so what we're starting to see here are genes that are known to be in the same pathways, and we're looking at the expression of these genes compared to the other groups, and how the set of genes move together in a direction or the other. And as you can see two types of patterns. These EGFR, NF-kappa-B, TNF-alpha, those ones, what you see is a kind of a situation where those pathways seem to be activated in that last group of combined treatment, potentially meaning that this is the recovery status that is still going on in these samples. Remember that this is after 5 weeks of having DMI, and so even if they're better in terms of the cardio phenotyping, they still are trying to recover, but successfully, apparently. There are other pathways, such as the MAP kinase and the P53, for example, where we're going back to the sham state in these analysis. So this is a lot of important data for the collaborators to look at and to try to understand further what these mean. Another analysis we can do is doing WGCNA. WGCNA is to look at correlation network in genes, and so very quickly, the genes that are co-expressed clustered together, we can group them by modules, and those modules can be also derived differently. There's many things that can be done using these kind of analysis. You can find the clusters that I'm calling modules. You can summarize them, relate modules either to each other or to traits, and also calculate module membership for each gene. And then when you do these modules, you can select some and go back. So here, it's not pathway driven. It's just, so the progeny analysis was pathway driven. It's known biology. We know those genes are working together. Here, we don't know they're working together. The only thing is we know that the expression patterns go together. So for example, this first, I think, magenta module is represented this way. So you might be able to see that there's lines here for the same genes going from one group to the other, and chronologically, it's not really chronological, but still we see that all these genes in the magenta module go back to the steady state or to the sham state. And again, with the yellow module, we have this opposite in this the same effect, but in the opposite direction, where they all get the expression has been higher during MI and other treatment, and then is going back to normal, to sham values in the combined treatment. So then we can go and look at those genes and try to do enrichment analysis of these genes, of these lists of genes. And in this magenta module, we've started seeing a lot of different things, but what caught our eyes was the mesenchymal cell development and all other stem cell development related pathways. And it's known that during this kind of the pulmonary hypertension of type 2, there's fibrosis, and there's also accumulation of stem cells. And so that was something that was known to happen, and that we can replicate and then see all the genes that would be involved in this process. Another pathway, so for example here, yellow module, we have a lot of other types of pathways that come out, and that was interesting because it's new biology that hasn't necessarily been found, and it's metabolism related, so we were interested in and the collaborators were very interested in this. And so, I'm showing you 2 examples, but there's very many examples to look at, and now it's a question of digging into the biology. Especially when we are seeing some things related to metabolism, because this is a picture of the metabolic pathways in humans, and so it's very, very complicated. It has a lot of different interactions, effects that can be happening, and this picture is actually incomplete. And this is also why it's very interesting to also go to this other omic topic, which is metabolomics. And this other project we have in collaboration with Dr. Marc Jolicoeur, Mathieu Ruiz, and also this work done by my bioinformatician Pamela. And so here we are studying specifically the metabolomics of myocardial infarction, and this time it's in humans. So it's actually an exploratory study of plasma metabolic profile. And we also have a drug response question here. So it's in a clinical setting, 180 individuals that have, for which we have acquired untargeted metabolomic data, meaning that we didn't specifically targeted metabolites that we chose. We just, so we, not we, but they, the company, generated data for metabolites in the plasma samples without the scientists being necessarily targeting some of these metabolites. In the clinical design, there was 3 time points at baseline, so people would have had a myocardial infarction, and they would have come either from 0 to 2 hours after the symptoms started, 2 to 4 hours, or 4 to 12 hours. And in each group, we had the same number of individuals, so 30, 30, 30. So it's 60, 60, 60 individuals for each of those time points. And then they were randomized such that the 2 drugs, for 2 drugs, the clopidogrel drug and the ticagrelor drug. Clopidogrel is the one that is usually given, and ticagrelor was the drug that they wanted to test. So Metabolon is the company that generated the data. And so once we received the data, we've had counts of metabolites for all these different samples, and obviously they're not yet cleaned of all the biases that can be found in biological and technical experiments. And so I'm not going to go through all of this either, but we've looked at different things. We've excluded samples and metabolites with too much missing data. We've imputed the ones that had still missing data after our threshold, our filters. We've done auto-scaling, and also in this case, we have the same individual in both groups before. So in the baseline group, so as they enter the study, and at discharge as they leave the hospital. So they're paired sampled, and we have to consider this. And as part of our quality control, we're looking at the data in different ways. So here you have an example of how the data was on a principal component plot before quality control and after quality control. So after quality control, we see neat separation, well, almost neat separation of these two baseline and discharge samples. We can try to look if we can find effect of timelines on these graphs, and we couldn't. And we can also look at whether at discharge we see an effect of the drug, and we couldn't. We're not so surprised over that, because principal component analysis is not necessarily meant to do these kinds of experiments. We've also looked at a new method to reduce dimensionality, but that doesn't so principal component analysis here, I'm only showing the two first principal component, but there's many of them that capture all the variability. UMAP is meant to capture this variability in 2D, and we were hoping to maybe start to find patterns in terms of the time points and the treatments, but I was not doing it either. So the same way as we did it for the expression data, for the gene expression data, we can use Volcano Plot, and this is inspired by our knowledge of the transcriptomics analysis. We can use Volcano Plots to look at the differentially quantified metabolites. So they're not expressed in this case, they're just the quantification is differential. And so what we see here is that, you know, most of the metabolites we have in our analysis are differentially quantified at this FDR, false discovery rate of 1% in our samples. So metabolomics is a very different, it has a very different landscape. It's a lot more dynamic. It has, it will shift a lot more than just gene expression, and it's actually closer to phenotypes than transcriptomic is. So that was an interesting aspect of looking at metabolomic data. We can, in the same way, do WGCNA analysis. So here you see all our metabolites, so no genes, now it's metabolites, and how they cluster together in different modules. We have less modules than what we had in the previous project. We have a big blue module here, a big turquoise module here, and a small brown module here. And what we can also do here, once we have these modules, and I mentioned it before, is to try to correlate these modules to traits. And here we wanted to see if we could correlate these modules to baseline and discharge status. And so this is what you see here. It might be very, very small, but this means that the blue modules is positively correlated with the status, the baseline status, and this one is negatively correlated with the baseline status. And in fact, if you look at this volcano plot, you should see that this is all those dots that represent metabolites in the turquoise model are on this side, so upregulated in baseline being the status of the myocardial infarction, and then all these blue module metabolites downregulated in the baseline status. We can have a look at what are the metabolites that are involved in these different modules. And the first one, the blue module, we're looking at different metabolites that are amino acids, mostly. And also in the turquoise module, most of these are fatty acids. It was pinpointed by our collaborator, Mathieu Ruiz, who's a metabolist, and who, for him, it was clear that all those metabolites in the are related to the fatty acid biosynthesis pathways. And so these are the main two components that are being changed in our data. Here, what we can look at is also for each individual, we can have a specific eigenvalue related to the blue module or the turquoise module. And here, so you have all the samples from the baseline, all the samples from the discharge, and you can see that most of them can be interpreted as down quantified in the blue module for the baseline and up regulated for the turquoise module. So this is another representation, but individually rather than the metabolites. So then when we do distribution of individuals, we can then try to look at the different traits or conditions we have. So we knew we had time points. And so when we look at the different distribution of individual eigenvalues for the different time points, what we've seen is a significant difference between, especially in the blue module, in terms of so for both it's actually significant, but we're seeing a difference in terms of the global metabolomic signature depending on the time where they arrived after the symptoms started. And so here we are seeing an effect before 4 hours and after 4 hours after the symptoms started. If we look at the same thing in terms of the treatment, unfortunately, we're not seeing anything interesting, anything significant in terms of these two blue and turquoise modules. But in our first models, we had used, actually, I forgot to mention, a three-way ANOVA model where we had interaction terms and where we were hoping to be able to capture interactions between the treatment and the baseline and the discharge status. And we've actually identified two metabolites. So it's not a huge number that are here, colored in pink, that are modulated by treatment. Another way of seeing them is this is the p-value for the treatment effect, and those two metabolites are significant, the rest is not. And then we could only take those samples at discharge because obviously at baseline they haven't had the treatment, so shouldn't see anything special for treatment. And we do the same thing with the WGCNA, but only at discharge this time. And so here you see this pink module coming out, which is moderately, weakly, significantly associated with the treatment. And when looking at where are those pink metabolites, we see those two that we've identified by an independent approach. And then we see other ones scattered across. We're actually not really sure they could be only correlated with those two, but without any effect. And Mathieu Ruiz, our collaborator, thinks that if there's a link, it will be quite indirect. So we're more confident about these two, and this is being followed up right now. So just to wrap up on those two projects, we've highlighted the transcriptomic signatures of a combined treatment that is highly beneficial compared to individual treatments in the case of pulmonary hypertension with PBI 40-50, we've discovered new metabolic pathways and processes involved in pulmonary hypertension following an MI. But the question that remains is, why does early treatment that was shown to be late treatment was shown to be very positive with only PBI. Why does early treatment with only PBI does not show the same effect? Is it possible that the valsartan component of the combination helps with going from this early to late status? In terms of the Metabolomics project, we found clear global metabolic signature of MI in human plasma with fatty acid enriched and amino acid depleted. That independently, these things have been suggested before, but what we have here is in human global signature, which is important. The type of treatment does not look like it influences the global signature. However, the time at baseline seems to have a moderate influence of the global signature. And so we're wondering now if we're looking at this immediate repair or a complete recovery, because we know that other studies have looked at the comparison between cases and control that had MI and have found some metabolic differences. We are in very early stages of after the MI, and it's unlikely that we are looking at a complete recovery. And so the last consideration here is that probably for studying all these interesting phenotypes and disease status, we would like probably to be able to combine transcriptomic and metabolomic for myocardial infarction. And in fact, in doing those two projects, we've seen fatty acid coming out in the transcriptomic, and vice versa. We've seen other interesting things crossing over the projects. And so even if here we're not specifically looking at these things, because we're in rats, we're in pulmonary hypertension, we're in humans, myocardial infarction, drugs are different, but it brings up the idea of combining all of this together, because in fact, it's not necessarily layered this way in the biology. It's more like a network of interactions between those different omics. I haven't talked about the genomics yet, but obviously different genes will different variation in genes will also influence transcriptomics and so on. And so it's very important to try to start to consider all of these together. And this is the newest part of my research. We're trying to get there because there's promising techniques in deep learning, for example, that are promising to us in terms of integration of data. Unfortunately, there's a lot of different challenges in that field. So first of all, the fat data challenge, where we have large piece or a large number of features, but a quite small number of cases in all our data sets. We have imperfect data sets, so I spent a bit of time with quality control. So this data is messy. It's also heterogeneous. So if we are looking at metabolomics, transcriptomics, genomics, proteomics, they all have their different particularities, and so it will be, it's a challenge to integrate all of this. And finally, the black box problem of these different machine learning approaches, where in the field of medical data science, we cannot just accept the result of an algorithm. We have to understand why this algorithm is giving a particular answer for a particular individual. And so there's lots of work that we need to do trying to interpret better these approaches. So with that, I would like to thank you for your attention, and I'm ready for your questions.
Donc, est-ce qu'il y a des questions pour Julie? Quelqu'un se lance? Oui, Sébastien.
The first study, I was curious, is the target of the drugs known? The drugs were tested in rats?
The PBI, it's kind of known. They're still testing the mechanism behind the drug.
Fine, so follow-up question is there any of these analyses that can help us identify what
Yes.
could be the target, and if so, how it could be?
Yeah, so we were kind of hoping that the PBI, the MI PBI group would give some results, at least to get into the biology of PBI alone. We can always lower our threshold to kind of try to, but we won't do this. However, given what we have on the combination and knowing what we know about the mechanism, so well, the company knows a lot more about their mechanism, so they might be able to tell us a bit about some genes specifically and identify the modules, and so there's a lot of digging that will need to be done in that area. But yes, the hope was, so it's partly funded by them, so the hope is to understand the mechanism of their drug. Yes.
For the second part of your presentation, was the target known as well, or?
Yes. Yes. So those are two. Yeah, it's known. I'm not exactly sure the.
Do you know if the results you have can confirm
Yes.
the target, or?
So for the drug, we have all these two metabolites, and right now there's no direct link to understand why those glycerol metabolites or glycerol-derived metabolites are linked to treatment, but it will be interesting to follow up.
Une autre question? Donc, merci Julie.
Merci.